Hi, and welcome to an introduction for the unknown report. During the course of your microbiology lab, you will be receiving images of an unknown bacterium. We have a list of organisms that we use in our face-to-face class, and we've chosen a subset of those to use in this online version. What will happen is you will each receive an assigned letter of an unknown bacterium, and then throughout the rest of the semester, well, most of the rest of the semester, you will be receiving images for that unknown in the lab archives notebook. The way I will organize it is there'll be a table with student names, and next to that will be the image of their unknown on the various media. You will only be responsible for recording the data for your own unknown microbe, along with both the positive and negative controls for each exercise. I have added this module of document for the unknown report to our Canvas page. The guidelines are here that go through everything you need to know about the report and how to write it. There is a document called the dichotomous key. And a dichotomous key means two cuts. Di meaning two and kato is to cut. Meaning we ask a question or do a particular physical or biochemical test, and the results are it does the thing we're looking for or it does not do the thing we're looking for. And this is how our key will start. We're going to do a gram stain, and all of the cultures on our list are either gram positive, so we would put the list of all the gram positives in this box, or they're gram negative, and all of the gram negative microbes would go in that box. You'll be provided with that information. There's no way I'm going to make you look up every single microbe and know all of its properties. Then from those that are gram positive, some of them have a rod or bacillus shape, and the rest of them have a coccus shape. Separate from this, of the gram negatives, some will have a rod shape, and actually none of the ones on our current list will have a coccus shape. Then the next question we ask for the gram-positive rod-shaped bacteria, do they have endospores or do they not have endospores? And for the gram-positive coccus-shaped bacteria, do they produce the enzyme catalase or not? Then for our gram-negative rods, do they use oxygen in a facultative way, meaning they can grow with or without oxygen, or are they a strict aerobe? And then since there were no gram-negative coccus-shaped bacteria, this whole side will no longer be relevant. Let's go back. The data, oops, let's not do the key yet. Let's look at the data for the dichotomous key. And these are the possible organisms: Bacillus cereus, Bacillus subtilis, Citrobacter frundii, E. coli, and going on and on. And then I provide you with which of those microbes are gram positive, which of those microbes are gram negative. Cellular morphology is the shape of the individual cells. These organisms have a coccus or perfectly spherical shape. These organisms have a rod or a bacillus shape. And now they are not divvied up by whether they are gram positive or gram negative. Bacillus and Bacillus are both gram positive. The rest of these are gram negative. So it's a separate test. You must keep that in mind throughout this process, that positive and negative are all independent answers for each test. Any organism can be positive for one test and negative for all the others, negative for the first test, positive for some, negative for others. Each test is independent. And then I have the oxygen use. We have only one organism that can only grow in the presence of oxygen. All of the rest can grow with or without oxygen. And only these two actually make endospores. And for the gram-positive cocci, some of them do make catalysts, some of them do not make catalysts. These will be explained in much greater detail as we get to those individual tests. But as we get into those, different students work at slightly different rates within each exercise. So I wanted to make sure that we had the data up so that people who are working a little bit ahead have the opportunity documents that they need. The last thing that's here in the documents for the unknown is the descriptive chart. This is where, in addition to each and every data sheet and questions for each exercise, you also need to record the results for your unknown on this chart. That's not just to make it easier on me, although it does do that. It's done primarily because it makes it easier for you. To identify your unknown, you don't want to be flipping back and forth through 20 different exercises worth of data. We record it there so that for each exercise we understand the biochemical properties and how the microbe works in relationship to the positive and negative controls. But we need this descriptive chart, so there's one place that you go to look for what did my unknown do on these tests. So it is organized by the morphological or shape characteristics, the cell shape, its gram reaction, how it appears in cellular arrangement, the various stains we're going to do, is it motile. And then we will have growth on a slant as one of the first times we'll see our unknown bacterium as growing on media, its oxygen requirements, and then we are going to be doing various metabolic capacities. And that's really the way that we can distinguish our bacteria. We can't just do it by shape. Millions of different organisms might have the same shape. That's not going to be nearly enough. The gram reaction helps. It cuts our possibilities in half, but that's still just one physical characteristic. We have to look at the entirety of their metabolism and the different types of capacity, and that's how we can separate them out. And so we have a way to record the information for all of the tests. Julie and I have organized this chart so that the way you record your information in this descriptive chart reflects the exact same way you recorded that information on your data sheets for each exercise. So make sure you record accurately on your data sheet and that you transcribe accurately from your data sheet onto your descriptive chart. As we move through the exercises, you will turn in through Lab Archives your individual data sheets and questions. The descriptive chart is your personal record keeping of all of your results. You should stay on top of it. And then there is a deadline for the completed descriptive chart. Once we've gone through all of the exercises, we have to get through exercise 5-24. Once we've done that, you will have all the data you need to identify your unknown, and your descriptive chart will be due. When you have that, we will have another video that will tell you how to use the dichotomous key and the more detailed dichotomous keys that are in exercise 9 of your lab manual.